Hi, my name is Gwendolyn Joy, and this is week's 2 video discussion on biofilm. Before we get started, I just wanted to go over my notes, just make sure that they are in the video. I also will let you know that I have my laptop open, but that is just so I can follow along with the questions, making sure I'm not just confused on what's on my paper, whenever I need to look down to make sure that I'm following along. But let's get started. Question 1 is asking, what are biofilms? And a biofilm is a complex complex community of organisms that are enclosed within an extracellular matrix. And question 2 is, what are some characteristics of biofilms and what makes them resistant to immune responses? And one of those key terms and just one of the characteristics is actually extracellular matrix, and its composition is a slimy, self-enclosed matrix, which is made up of proteins, DNA, as well as polysaccharides. And those create and act as a barrier, making it harder for the antibiotics or anything else to kind of penetrate and get through them. And another thing that makes it harder to be penetrated through is the parasitic cells. And those are hard to penetrate because of the fact that they have a slow growth rate. And that slow growth rate causes them to kind of go into an inactive stage, creating the antibiotic to be able to disperse freely. And it's not able to pick up on that bacteria. And because it's now creating a high tolerance and resistance for those antibiotics. As well as slow growth rate is actually another key character term for biofilms. And because of the slow growth rate, again, the antibiotics cannot target and look for those active dividing cells. And because there's nothing that they can kind of locate, they just kind of disperse. And again, it just allows the biofilm to stay firm. Some other key terms for biofilms are the effectiveness of antibiotics that target, physiological heterogenicide, quorum sensing, efflux pumps, and alter target sites. Now we're going into question 3, which are, what are polymicrobial biofilms and what are some examples? And they are the microbial biofilms are communities of microorganisms that grow in a coordinated manner. And some examples of those are dental, wound, and biofilms that are also can be on medical devices as well as lungs. Going into question 4, many antimicrobial biofilms interventions are studied using single species platonic microbes. Why are these insufficient? Why is this an insufficient approach? And this is because the antimicrobial biofilm actually has the biofilm has a complex community of multi-species bacteria, which they all interact, causing it harder for, which interact within the biofilm, which now creates a way for it to be harder to interact, which then causes a higher tolerance rate, to the higher resistance to the antimicrobial, which is composed of isolated single species bacteria. Some examples is how it increases the resistance of multi-species biofilms, physiological heterogeneity, I can never say this, of heterogeneity, biofilms matrix complexity references to the chemical affirmations. And to just kind of, there we go. And just to coordinate of what that means is just because it can't interact and communicate, it's causing this blockage. So it just can't communicate and be able to coordinate of how do we break through or what do we target. And now leading us into our final question for part A is, describe and provide examples of the following tactics currently being developed, implemented to prevent erratic biofilms. Our first one is antimicrobial peptides, which are small proteins that defend microbes. And by defending the microbes, one of the examples is a BMAP or BMAP 28, which is a bovine AMP that permeabilizes the membrane of numerous bacterial fungi. And some of those are just kind of ways of just saying like, oh, it can't get into the microbes, which is great because then it can't penetrate and then still break into those barriers, which then can build up for the biofilm. Another biofilm that you wanted us to talk about is a dispersal methodologies. Dispersal. We're learning big words this year. And those are how, it's a study of how the organism can change its location, so how it relocates itself within the cell. And some of those methodologies or mechanisms is through gravity, water, wind, as well as ballistic and animal. And the way that those are kind of all tolerated and coordinated in is through water. It goes into our streams and it disperses because of the riptides and the flow of just the stream itself and the flow of gravity. Through animals, because when the animals either are eating them and it disperses through their fecal matter, or because it gets attached to their bodies and then when they go around, that's one way, or again just by eating and consuming, or the storaging portion. And another example of is just the wind picks it up, takes it away. And then for the plants, another way that the plants do it, which kind of coordinates with our wind as well as animals, is that they'll do kind of an explosion or self-induce kind of disperse, like dandelions, because of the wind. It takes those and it kind of disperses itself. Another is the EPS interdiction, and that is the polymers, the EPS guaranteed by their living organisms. This antimicrobial bacteria, tumor, and immunology effects, which is kind of honing in on their properties like microcompatibility, holy graham crackers, microcapability, degradability, learned how to read, and the lack of irritation. And these are actually all stopping, and these are kind of blockers of, again, the tumors, GSMs, bacterial and antiviral. So those are canceling out all of those huge, big no-nos in our life that we don't want. And then we also have nanotechnology, which is the use of techniques of nanomaterials to study, alter, and microorganisms at the molecular level. And these are environmental remediations, diagnostic tools, drug delivery, immunology, Devol's techniques. Some of their techniques are the imaging, manipulations, and spatula organization. And then lastly, we have our phage therapy as well as the quorum quenching. But before we get to those two, I do want to apologize because I know I'm going to be going over the time limit, but promise I'm almost there. Again, so one of our other biofilms that I will be describing is phage therapy, and phage therapy is this technique that is used to kill off bacterial phages. That uses I think I just treatment of bacterial infections that uses viruses called bacteriophages. I was like, that's not what I meant. And those are to kill off the bacteria. And some examples is how the phages are genetic material. It makes clones of itself, and that way it's harder to inject. And so those so the bacteria cannot actually, so those bacteria cannot kind of reproduce because it's just making a clone constantly of itself, and so the bacteria can't function and create more bacteria. And then lastly, we have our quorum quenching, which it interferes with the quorum quenching. So sorry. Jeez,